Welcome to everybody. Today in this video we will discuss about prokaryotic cell and eukaryotic cell and the differences between prokaryotes and eukaryotes. There are two types of cells. The first one are the prokaryotic cells and the second one are the eukaryotic cells. Now, prokaryotic cells are seen in kingdom Monera and eukaryotic cells are seen in kingdom Protista, Plantae, Fungi and Animalia. So before we discuss about the difference between prokaryotic cells and eukaryotic cells, let us try to understand the characteristics of these two types of cells. So prokaryotes, I'll divide this word prokaryotes into two words, pro and karyon. Pro meaning early or before and karyon meaning nucleus. So prokaryotes meaning before nucleus. Prokaryotes are the most primitive cells. Most primitive cells meaning they are the most ancient cells. And prokaryotes are said to be evolved 3.5 billion years ago. Prokaryotic cells are enveloped by a single membrane system. Now, this single membrane system is also known as cell envelope. And this cell envelope comprises of three tightly bound layers: glycocalyx being the outermost layer, cell wall being the middle layer, and plasma membrane being the innermost layer. The genetic material is not enclosed by any membrane, so it lies in contact with the cytoplasm. So I'll take the most common example, bacteria, and for example, this is your genetic material, and this genetic material is not enclosed by any membrane and it is lying in contact with the cytoplasm. Okay, this is the cytoplasm and the genetic material is lying in contact with the cytoplasm. This is your genetic material and that is the cytoplasm, and there is no membrane which is surrounding it. So the region where the genetic material is present is referred to as nucleoid. Most of the people get confused that the genetic material of prokaryotes is known as nucleoid, but it is not nucleoid which is the genetic material of prokaryotes. It is the region where the genetic material of prokaryotes is present, since the genetic material lies in contact with the cytoplasm. And the region where the genetic material is present is known as nucleoid. So we need to remember that. Moving on, membrane-bound organelles, such as ER, Golgi complex, mitochondria, they are absent. Complex cell organelles or membrane-bound organelles, they are absent. Membrane-less organelles are present. Ribosomes are present in prokaryotes, and they are 70S type. Now, what is the most important thing that we need to know are the examples. The examples of prokaryotes. When I think of prokaryotes, these examples should come to my mind. So the most common example of prokaryotes is bacteria. Whenever I think of prokaryotes, the first example that pops in my mind is bacteria. Cyanobacteria, which is also known as blue green algae, and PPLO meaning pluro pneumonia like organisms. So organisms and mycoplasma, which is the smallest among all of these. Now, when I think of prokaryotes, what needs to come to my mind is genetic material, not enclosed by any membrane. Secondly, membrane-bound organelles absent. Third, which could be which is there, is that their organization is very simple. Okay, and then the examples of the prokaryotes. Moving on to the second type of cells are the eukaryotes. So I'll break down this word into two words, eu and karyon. Eu meaning true and karyon meaning nucleus. Eukaryotes, they have a distinct nucleus, that is, the hereditary material is enclosed in a double membraned nuclear envelope, and this nuclear envelope is also known as nuclear membrane. Okay, so for example, if I take an animal cell, and for example, this is your genetic material. So this is your double layered membrane, which is also known as nuclear envelope. Okay, so we could see that there is a distinct entity, and in this distinct entity, that is nucleus, is the hereditary material or the genetic material enclosed, which was not in the case of prokaryotes. It was lying in contact with the cytoplasm, but here we could see an entity. Okay, so the genetic material is not lying in contact with the cytoplasm, and we could see that it is different from the cell. I could see that this genetic material is surrounded by a boundary which differentiates it from the cell as well, right? So distinct nucleus means that it is recognizably different. I could see it. I could see that, this is cell, and in the cell entity is present, which we call it nucleus, and in this nucleus genetic material is present. Secondly, membrane-bound organelles are present, like mitochondria, endoplasmic reticulum, vacuoles, and lysozymes. It doesn't mean that they do not have ribosomes. They have ribosomes. And ribosomes are of ATS type. Okay, so when I think of eukaryotes, you could think of animal cell, plant cell, in protists even, eukaryotic cells are seen, in fungi even. So eukaryotes, what are the characteristics of eukaryotes is simply that they have membrane-bound cell organelles, and their nucleus, their genetic material is enclosed by a nuclear membrane. Now, a network of endomembranes, okay, there is a network of endomembranes which will divide the cytoplasm into channels, which means it will divide the cytoplasm into compartments. When we will study about the cell chapter, we will try to focus on this point, and we'll see how, it is this network of endomembranes is dividing the cytoplasm into channel. It is basically the endoplasmic reticulum. So we'll see it in the chapter cell. And a eukaryotic cell can be broadly separated into cell membrane, cytoplasm, and nucleus. Okay, whereas in the case of prokaryotes, it is broadly separated into cell membrane, cytoplasm, and genetic material. In that, there is no nucleus present, which we need to keep in mind. Moving on, let us know the differences, okay, between prokaryotes and eukaryotes. Okay, so the first feature which we will discuss is the cell wall. Okay, in prokaryotes, it is made up of numeric acid and amino acids. In eukaryotes, it is absent in animal cell and it is present in plant cell, and it is composed of cellulose. Fungi's cell wall is made up of chitin. Okay, and it is made up of numeric acid and amino acids in prokaryotes. In eukaryotes, it is made up of cellulose if it is seen in plant cell. In animal cell, it is absent. Right? In fungi, it is made up of chitin. So, moving on, plasma membrane. They are made up of phospholipids. Here also the plasma membrane is made up of phospholipids, but the phospholipids it is made up of is known as haploids, and here it is known as cholesterol. The chemical composition is different. Okay, they're made of phospholipids, but phospholipids can be of also various types. So, the phospholipid which make the plasma membrane of prokaryotes are known as haploids. And in the case of eukaryotes, the phospholipids which make the plasma membrane is called cholesterol. Okay, so there is a thing that an outer covering is needed for protection. In animal cell, there is only plasma membrane. In plant cell, there is cell wall and plasma membrane. Okay, in prokaryotes, if I talk about bacteria, there are 3 layers present. We discussed glycocalyx, plasma membrane, and cell wall are present. The chemical composition of the layers is different. They have this protective covering. There is a protective covering in eukaryotes as well as in prokaryotes, but it is different for different cells. Okay, and the composition is also different. So we need to keep the composition in mind, because sometimes they ask these things about the composition, that the cell wall of prokaryotes is made up of, and they would give you options, and we don't know about the chemical composition of prokaryotes or eukaryotes. We just know that in eukaryotes it is absent in animal cell and present in plant cell, and it is composed of cellulose. We basically don't know about what is the composition of cell wall in prokaryotes, right? Nuclear membrane, absent, present. We discussed at this point earlier that there is a nuclear membrane present in eukaryotes, but it is absent in prokaryotes. And the nucleoplasm is basically, the cytoplasm which is present inside the nucleus. So since there is no differentiation from the cytoplasm, the genetic material is not differentiated from the cytoplasm, there is a nucleoplasm would not be present, and it is not differentiated. Basically, it is the cytoplasm which is present near the genetic material. So in prokaryotes, it is not differentiated from the cytoplasm, but it is separated by a nuclear membrane from cytoplasm in eukaryotes. Nucleolus is absent. It is present in eukaryotes. Now, chromosome. This is a very important point. In prokaryotes, there is a single circular structure which is formed of DNA. Here, it can be more than one, and it is composed of DNA. Okay, here a single circular structure is present, but there are no basic proteins, histones, whereas in eukaryotes, basic proteins called histones will help in the binding of the DNA. Okay, in prokaryotes, there are different type of proteins present, but we'll discuss it when we'll talk about bacteria. But histones, which are the basic proteins, they are not present in prokaryotes. Okay, moving on, ploidy. Ploidy, what does the word ploidy means? It means the set of chromosomes. Okay, most of the people, they know what is haploid and diploid, but they actually don't know what it means, right? So haploid is basically this condition. There is only one set of chromosome present. Okay, so that is why they are referred to as haploid. and diploid is this condition where two sets of chromosomes are present, which is seen in the case of eukaryotes. Okay? Cell division, simple fission. Because there is no formation of spindle fibers, so no mitosis and meiosis are going to take place. In prokaryotes, it is simple fission. Now, the simple fission could be multiple fission, could be binary fission, could be anything, but since spindle fibers are not formed in it, so no mitosis and meiosis is going to occur in this. In eukaryotes, cell will divide by mitosis and meiosis because spindle formation take place. Moving on, transcription and translation. Okay, we know in prokaryotes the genetic material lies in the cytoplasm. Okay, so all the chemical reactions are going to take place in cytoplasm. So transcription and translation is also going to occur in cytoplasm. But in eukaryotes, transcription will occur inside the nucleus and translation will occur inside the cytoplasm. Respiratory enzyme. This is a very important point because respiration is a very important process. Respiration is the process by which we obtain energy. Okay, so these respiratory enzymes, where they are present. Okay, so in eukaryotes, we know that these respiratory enzymes are enclosed in mitochondria, and that is why it is also known as the powerhouse of the cell. And this is responsible for generating your ATP, right? So that is why it is also known as the powerhouse of the cell. Now, since we know in prokaryotes, membrane-bound organelles are not present. Mitochondria is a double-membraned organelle. Okay? Respiratory enzymes. Now, what happens is that this is your plasma membrane, and you would see infoldings in it. And when these infoldings or invaginations, okay, are seen in plasma membrane, we refer it to as mesosomes. Okay, it is basically an infolding of plasma membrane, and इसके ऊपर respiratory enzymes present होते हैं, which will help in the process of respiration. Okay, they will be responsible for making energy for the prokaryotic cell, because इनमें membrane bound organelles नहीं होती, तो इनफोल्डिंग हो जाती है प्लाज्मा मेम्ब्रेन की, और इस इनफोल्डिंग ऑफ प्लाज्मा मेम्ब्रेन को हम एक स्ट्रक्चर की तरह ट्रीट करते हैं जिन्हें हम मेसोसोम्स कहते हैं, और इन मेसोसोम्स के ऊपर या इस इनफोल्डिंग ऑफ प्लाज्मा मेम्ब्रेन के ऊपर रेस्पिरेटरी एंजाइम्स प्रेजेंट होते हैं, और ये these are responsible for respiration. Okay, so these are some of the most important differences that we need to keep in mind.
Thanks for watching this video. Please don't forget to like, share and subscribe everybody.